Recombination in bacteria. The chromosome of bacteria, unlike those of eukaryotic cells, are not enclosed in a nuclear membrane. The bacterial chromosome contains a single circular double-stranded molecule of DNA which occupies a definite position in the cell cytoplasm. The nuclear region of bacteria is often referred to as nucleoid. In bacteria, the genetic information is mainly stored in the single main chromosome. Besides this, the bacterial cell contains several extra chromosomal structures called plasmids which also contain genes. Certain bacterial cells harbor as many as 11 different plasmids. Recombination is an integral part of sexual reproduction in higher organisms. This is the production of new gene combination not found in parents by the crossing over between homologous chromosomes during meiosis. Recombination therefore brings an array of variations which form a raw material for evolution. However, in bacteria, reproduction is mainly by simply binary fission and bacterial cell is haploid, having only a single set of genes. Therefore, bacterial cell cycle is not characterized by the usual recombination events associated with the sexual reproduction in higher organisms. However, recombination of a different sort take place in bacteria too. It involves the transfer of genetic material from one bacterium to another, making possible the subsequent recombination events. Recombination takes place in bacteria by three different processes which differ in the mode of transfer of DNA from one cell to another. They are transformation, conjugation, and transduction. So, transformation involves the uptake of free naked DNA molecule from one bacterium or donor to another bacterium or recipient. The first evidence of bacterial transformation was given by Griffith in 1928 during his studies on the pneumonia-causing bacterium Streptococcus pneumoniae, which led to the understanding of DNA as the genetic material. During his studies, Griffith discovered that when new virulent rough strain bacteria were cultured in the presence of heat-killed virulent smooth strain, the rough strain was transformed into smooth strain. Avery, MacLeod, and M.C. McCarthy in 1944 identified that the transforming principle was DNA, which could be extracted from donor strain in pure form. Destruction of bacterial cell does not necessarily destroy its genetic material. When bacteria live, they often release their DNA molecule into the surrounding media. This DNA retains its ability to synthesize specific proteins if introduced into another bacterial cell. Recipient cells absorb fragments of donor DNA onto their cell surface. The DNA is then transported across the cell wall and plasma membrane and is incorporated into the recipient's chromosome by recombination. The recipient cell now acquires new characteristics. This process of bacterial transformation takes place in three successive stages: competence, binding and uptake, and integration. So here we could see a picture which depicts this process. First, recipient DNA is there. So A plus double-stranded donor DNA. One strand of donor DNA enters cell as the other gets degraded. Then formation of triple strand and double crossover recombination by strand displacement. Recipient DNA segment with A allele degraded. Then chromosome with segment A plus A heteroduplex DNA duplication taking place. Non-transformant, one transformant. All cells in a given population are not capable of active uptake of DNA. Only competent cells can do so. Competence is the ability of recipient bacterial cells to take up DNA through changes in the cell wall. There are special DNA receptor proteins on the surface of cell wall of these cells. Competence involves either the formation or activation of these receptors. The number of these receptors varies from species to species. Next, that is competence. Then binding and uptake. This involves direct interaction between the cell wall receptors and fragments of donor DNA, which is followed by the uptake of donor DNA into the recipient cell. In the gram-negative bacteria, Haemophilus influenzae, the donor DNA is taken into the recipient cell as double helix. In the gram-positive bacteria like Streptococcus pneumoniae and Bacillus subtilis, the DNA double helix is first nicked by endonuclease to form small piece of double helix. One of the two strands of the double helix is then digested by an exonuclease to yield single-stranded molecule which are absorbed into the recipient cell. Then in integration, the stage begins with the intracellular transport of the transporting DNA to the recipient chromosome in some protected form, either complex with a specific protein or in some vesicle derived from the cell wall of the recipient cell. If the donor DNA is taken into the recipient cell as a double helix, as in Haemophilus influenzae, it is converted to single-stranded molecule by nucleolytic degradation of one strand. During integration, this single strand is incorporated into the recipient chromosome by displacing a homologous section of one of the recipient DNA strands, which is then excised and degraded. This results in the formation of a transient heteroduplex. During the next cell division, the incorporated donor strand acts as template for the synthesis of a complementary strand, thus resulting in a completely transformed cell. So conjugation, it was Liderberg and Tatum, 1946, who discovered sexual conjugation in E. coli. Conjugation is a one-way transfer of DNA from the donor cell to a recipient cell through a specialized intercellular connection called conjugation tube. The donor, sometimes designed as male, differs from the recipient female in the possession of an extra chromosomal plasmid known as X factor or fertility factor or F factor. F factor is a circular double-stranded DNA with a molecular weight of 35 into 10 raised to 6 and having about 100,000 base pairs. Bacterial cells with F factor are called F positive, and those having F factor without F factor are called F negative. Conjugation always takes place only between F positive strain and F negative strain. During conjugation, the F positive cell readily establishes contact with the F negative cell through a long appendage that extends from the surface of the F plus cell to the F negative cell. This appendage, called F pilus, later gets modified as a protoplasmic channel called the conjugation tube between the two cells. The formation of F pilus is controlled by the F gene. Mechanism of conjugation. During conjugation, the only genetic material that is transferred from donor F plus cell to recipient F minus cell is the F factor. Each F plus cell will transfer a copy of its F factor onto the F recipient while retaining at least one copy for itself. The F minus cell that receives an F factor is transformed into F positive cell. The transfer of F factor from F plus to F negative cell is coupled with DNA replication by rolling circle mechanism. A cut is made in one of the two strands of DNA double helix of the F factor, and the cut is drawn to the F negative cell through the conjugation tube with 5' end of the front. The rest of the DNA strand follows sequentially in a linear order. The other strand of F factor DNA remains in the donor cell. In the F cell, the newly drawn single strand acts as a template for the formation of its complementary strand at the 5' 3' direction. The single strand of F factor that gets remained in the donor cell serves as a template for the synthesis of the missing strand. Thus, at the end of conjugation, both the conjugates will contain a copy of F factor and they separate. Now, both the donor and recipient cells are F positive cells. Look at this picture. F plus male, F negative female. One strand is transforming to next one. Conjugation in bacteria. The F factor can exist in two different states. In the majority of F positive bacterial cells and F negative, F factor remains outside the bacterial chromosome as an autonomous cytoplasmic element. However, in certain F positive cells, the F factor becomes integrated with the main bacterial chromosome, and such F positive cells with integrated F factor are called HFR cells. HFR for high frequency of recombination. Since F factor is capable of integrating itself into the bacterial chromosome, this plasmid is also referred to as the episome. In the integrated state, the F factor mediates the transfer of a chromosome of the HFR cell to a recipient F negative cell. The integration of F factor with the bacterial chromosome takes place very rarely. Occasionally, the F factor may leave its chromosomal portion in the HFR cell and the cell gets converted to H positive cell. The sites at which integration between the F factor and the bacterial chromosome occur as specific segments of DNA is known as insertion sequences. In HFR cell, and F factor is replicated passively along with the replication of bacterial chromosome. During conjugation, the F factor transfers part of itself and part of all one of the bacterial chromosome strands to the recipient cell. If chromosome transfer by the donor cell is complete, the integrated F factor is transmitted along with the bacterial chromosome. Then the recipient chromosome recombines with the F portion of the donor chromosome and gets converted into HFR cell. If transfer is incomplete, the recipient cell may not get the F factor and still remain in the F negative condition. The donor chromosome that is in the process of transfer to recipient cell may break anywhere along with its length, and in such cases the transfer is incomplete. The breakage occurs randomly throughout the length of the chromosome. Thus, longer the chromosome that is being transferred, the more points along its length that are breakable, and the greater the chance of incomplete transfer. During complete transfer, the F negative cells receive the F factor as well as the entire set of homologous genes from the HFR donor cell. The transferred cell is now said to be diploid zygote. However, during incomplete transfer, only part of the donor chromosome is transferred and the recipient is diploid for some donor and recipient genes. Under such circumstances, this is called partial zygote or mesozygote. Transduction is the virus-mediated gene transfer in bacteria discovered by Zinder and Leidenberg in 1952. It occurs when a bacteriophage particle carries a segment of the chromosome from one bacterium or the donor to another bacterium or the recipient, facilitating the subsequent recombination of the genetic elements of the two cells. Bacteriophage and Bacteriophages are viruses that infect bacteria and which bear a parasitic relationship to their hosts. The phages multiply inside their host cells, leading to the destruction of or lysis of the host cell. Such phages are called virulent or lytic phages. However, some phages maintain a symbiotic relationship with their bacterial host without causing immediate lysis. Such phages are called non-virulent or temperate phages. The DNA of the virulent phage remain outside the bacterial chromosome and divide independently. On the other hand, the DNA of the temperate phage is integrated with the bacterial chromosome as a prophage and replicated in synchrony with the bacterial chromosome. The bacterial cell which harbors a prophage is known as lysogenic because under appropriate conditions the prophage can leave the bacterial chromosome, divide independently and enter into the lytic cycle. Lysogenic bacteria are immune to secondary infection by the same viral species. Transduction can be mediated by virulent phages as well as by temperate phages. Transduction mediated by virulent phages are called generalized transduction, and one mediated by temperate phages is called specialized transduction. Generalized transduction. All virulent phages do not mediate transduction. E. coli phage P1, Salmonella phage P22, Bacillus subtilis phage PBS11 and SP10 are some generalized transducing phages. During their lytic cycle, these phages often commit mistakes. Usually during their lytic cycle, phage chromosomes replicate repeatedly to produce a large number of copies, and each phage chromosome is packaged into a protein coat to form a complete phage. However, bacterial genes can become accidentally enclosed within a bacteriophage during the lytic cycle of the phage inside the host cell. This happens because during the lytic cycle, the host chromosome may be degraded into small pieces and occasionally a fragment. This happens because during the lytic stage, the host chromosome may be degraded into small pieces and occasionally a fragment similar in size to the recipient replicating viral chromosome is packaged into the viral protein coat. These viral particles with bacterial genes are called transducing particles, viral particles that contain bacterial genes. These are liberated along with normal progeny viruses during the lysis of the host cell. When a transducing particle subsequently infects a susceptible host cell, the genes from the previous bacterial host are introduced into the new cell. Because viral genes have been replaced by bacterial genes, so new bacteriophages are produced. But the newly introduced bacterial genes may replace the homologous segment of the bacterial chromosomes. Since any portion of the bacterial chromosome can be transferred, this type of transduction is referred to as generalized transduction. Infection of donor, lysis, section of bacterial chromosome in virus, infection of recipient, pairing, incorporation of donor section, events in generalized transduction. Then next, specialized transduction. Specialized transduction is mediated by temperate non-virulent phages whose DNA is integrated with the bacterial chromosome. Temperate phages have two portions. Either they can remain integrated with the bacterial chromosome as a prophage, and replicate in synchrony with the replication of bacterial chromosome, or they can separate from the bacterial chromosome to enter the lytic stage and start autonomous replication. Most of the temperate phages like coliphage lambda occupy specific or fixed positions in the bacterial chromosome called prophage site. So phage DNA, A, B regions, donor bacterial gene and donor bacterial gene are located. Then free phage DNA with bacterial gene, DNA of recipient cell cleaves, transcription happening, then recipient gene, phage DNA, recipient gene, donor DNA, mechanism of specialized transcription. Temperate phages undergo rare spontaneous transitions from the prophage state to the lytic state, and such transitions can be induced by UV radiation. When a prophage disintegrates from the bacterial chromosome, it accidentally picks up a new adjacent bacterial gene also. This phage host DNA hybrid replicating autonomously and become packaged into the transducing viral particle. When they infect a host cell and integrate into a chromosome, the recipient cell acquires the characteristics specified by the newly introduced genes of the previous donor host. This type of gene transfer is called specialized transduction because only those genes that lie close to the phage integration site can be transferred. The integrated F factor in the HFR cell may exit the bacterial chromosome by reversing the steps that resulted in its integration. Sometimes there is an error in the excision of the F factor from the chromosome. This error results in the formation of an F negative factor, F dash factor, factor containing a segment of the bacterial chromosome. Meeting of an F dash factor with an F negative cell basically is same as F positive into F negative. With one important difference, bacterial genes incorporated in the F plasmid are transferred with very high frequency to recipient cells. These genes do not have to be integrated into the recipient cell's chromosome to be expressed. The recipient acquires the F negative factor and is generally diploid for the genes that it carries. The transfer of bacterial genes by F negative factor is called as extraction. Extraction results in the rapid spread of bacterial genes from the main chromosome through a population. So that's all about this topic. We just had a quick recap about this topic of recombination in bacteria.